Yes,
See the
ma'am.
screen? Okay.
It is showing on the first page, ma'am.
Yeah. Now you can see?
Yes,
Yes,
ma'am.
ma'am.
Okay.
Ma'am,
Now coming
it
to
is
cell organelles.
it,
Now coming to cell organelles of the cell. Just one
ma'am,
second.
it is showing.
Yeah, now you can see the screen, I hope. If you can't see, please log in again. Join again, I'll admit you. Join again, I'll admit you. So coming to cell organelles of the cell, children. There are different types of cell organelles present in an animal cell. So don't get confused if I say animal cell, because for animal cell and human beings, the cell organelles are same. But for plant cells, there is a difference. Because for plant cells, there are chloroplasts, which are helpful for them for photosynthesis. There are extra organelles in plant cell, like cell wall also. Whereas the animal cell do not have a cell wall, a rigid cell wall, because there are so many functions to be carried out by an animal cell. Like there is an exchange of different types of materials in and out of the cell. So children, here comes the mitochondria. As we all have learned earlier, that mitochondria is also called as the powerhouse of the cell. Mitochondria are the oval structures. There are more than one mitochondria present inside the cell. Mitochondria have its own DNA, genetic material, through which it can replicate itself. So these mitochondria play an important role in releasing the energy. It just break down, it helps in breakdown of the glucose molecules by producing the energy. So the energy currency is called as ATP, adenosine triphosphate. So you'll be learning about it in your higher classes. So now comes next organelle, which is called as Golgi body. Golgi body, Rudrinish, I have just started just now. You haven't missed anything. I have started about the cell organelles. If you are raising the hand for the sake of asking about what you have missed, yes, you can speak. You have any doubt? Rudrinish?
Ma'am, did I miss any?
No, no, you haven't missed anything. You haven't missed anything. I have started just now. Okay. So I have just explained about the mitochondria. So mitochondria is called as the powerhouse of the cell. These are spherical oval structures, or they also rod-shaped organelles with a double membrane. They have inner foldings inside them, which are called as cristae. So these mitochondria generate energy. They break down glucose molecules and they produce energy. So these are the energy currency which is being produced inside the mitochondria. It's called as ATP, adenosine triphosphate. It is called as the energy currency. So this energy is being utilized by the cell. Now coming to Golgi body. So Golgi body is a flat, smooth layered. I will just copy this image here, so that it will be easy for you to understand. Just one second, children. If the picture will be beside this, so it will be easy for you to understand. Just one second, children. Okay, one second. I hope you all can see it. Okay. So now look at this picture. So this Golgi body, can you look at the Golgi body here inside the cell? Yes, it is here. This is the Golgi body. So this is a flat, smooth surface. It doesn't have any rough surface. It has smooth layers. So it is a smooth layered organelle, sac-like organelle. It appears just like a sac, a bag that is located near the nucleus. This is the nucleus and it is nearby. It is located nearby the nucleus, having enzymes and proteins. This Golgi body gathers simple molecules and combines them to make molecules that are more complex. Just once again, children, I think the students, they are attempting to join. Okay. Yes, students. So now coming to vacuoles. What are vacuoles? Vacuoles are different. They perform different functions in different types of cells. Like they help in removal of waste, and at times they also store food material in some sort of cells, or any variety of nutrients the cell may need to survive. They even provide, they even store the food, and in certain type of cells, at times, they even provide nutrients if the cell is in a need to survive. Yes, where are these vacuoles located inside a cell? This is an animal cell, and this is a small vacuole which is present inside a cell. There are one or more than one number of vacuoles. Here you can locate one more vacuole here and one more here. Because it is an animal cell, the animal cell have more than one vacuole. There are one, two, and three number of vacuoles because they are small. But in plant cell, where is the vacuole and how it is? It is very big, isn't it? The vacuole is very big inside a plant cell. It covers more than 80% of the cell area. Okay, children. So now coming to, just once again. Yes. Vacuole. So I hope you all have understood about this vacuole. Now coming to endoplasmic reticulum. I'll just move this image over here. Just one second, children. Yeah. Yes. So now you can see the endoplasmic reticulum. Where is this endoplasmic reticulum here? You can see it here. This is the endoplasmic reticulum, which is being written as ER. This is rough ER and smooth ER. ER is nothing but endoplasmic reticulum. This here we have smooth endoplasmic reticulum and rough endoplasmic reticulum. Do not get confused with the name, children. ER is nothing but endoplasmic reticulum, because on certain endoplasmic reticulum there are ribosomes which are being attached. The red color dots which you can see on this endoplasmic reticulum, so it is called as rough because the ribosomes are being attached there. So because of that, it is called as rough endoplasmic reticulum. And to the endoplasmic reticulum on which the ribosomes are not being attached is called as smooth endoplasmic reticulum, because nothing is being attached there. So I'll just show you once more the video for this cell organelle so that it will be clear to you. Yesterday we have seen only half of the video. So I'll just show you the rest of the part also, or from the beginning also. So we'll be having a clear view on the centrioles and lysosomes also, children. So what is the function of this endoplasmic reticulum? Endoplasmic reticulum has a ladder-like appearance. It connects various cell organelles and transport the nutrients from one cell part to another. They have a beaded appearance due to the presence of ribosomes. Beads are nothing but the ribosomes. Now coming to centrioles and centrosome. So where is the centrioles present? So these centrioles are present in uh, animal cells. So I hope it's not found here because it's not mentioned here. So what are the functions of these centrioles? Centrioles are present in animal cells. A centriole is made up of a pair of centrosome is a small organelle found close to the nucleus that has a thick center with radiating tubules. Centrioles and centrosome are active during cell division. So now coming to lysosomes. When these centrioles are being seen during cell division, when cell is about to divide, the nucleus is also dividing. When this cell division takes place, then we can see these centrioles and centrosome. Otherwise, we cannot find them. They are present near to the nucleus. Now coming to lysosomes, children. Lysosomes. Why Harsh have joined so late? I think he's having some problem in the internet connection. Lysosomes, children. Lysosomes are also called as suicidal bags. They are called as suicidal bags. Why they are called as suicidal bags? Because they are filled with enzymes that can dissolve the cell. They contain enzymes, powerful enzymes, which are capable of dissolving the own cell itself. If by accidentally this lysosome breaks, then the cell may die because of the very powerful enzymes which can digest themselves. Lysosomes are cell organelles which clean up the cell. What is the function of lysosome? They clean up the cell by digesting the foreign materials which is being taken, which gets into the cell. Either it might be a bacteria also. When an invader is there, children, like a bacteria, suppose a bacteria enters into our body, then what the cell do? The cell will engulf it. The bacteria will first go and attack the cell. It enters into the cell. Then what the cell do? The cell will first acts upon it because it's a foreign body. Then first the vacuoles just ingest them, and they make the bacteria kill with the help of powerful enzymes. Okay, children, I hope you've understood. So we'll be having a clear view of the, I hope you all can see, children. Switch on your camera so that I can see you all. Yes, you all can see it or not? Is it visible to you? Okay, that's fine.
Everything is managed from here. Don't miss the power station. It keeps the factory running. This is where all the work is done on machines. Did you know that just like this factory, there are tiny factories in our bodies as well? Now, sit back and take a trip of the factory running in our bodies. By the end of this lesson, you will be able to identify the parts of a cell, identify different cell organelles, and differentiate between plant and animal cells. Hi, I'll be your guide through the rest of the tour.
You all can see it? No. Srinika, you can join again. Others you can see? Everyone, Manogna, Vilohit, you can't see? Okay, I'll share it again.
It is a living membrane made of lipoproteins. The plasma membrane is selectively permeable as it allows the movement of some substances in and out of the cell. That's my selectively permeable plasma membrane. I'll show you the different ways in which substances move through it. There is spontaneous movement of gases like carbon dioxide and oxygen from a region of high concentration to a region of low concentration to form homogeneous solutions through diffusion. The diffusion of solvent molecules through a semi-permeable membrane from a region of low concentration to high concentration is called osmosis. Let's try this out. See what happens when the cell is placed in salt solution. One of three things could happen now. When the cell loses water through important hereditary information and are composed of deoxyribose is the control center of the cell, called this is known as plasmolysis. Let's now look into the cell. This is the control center of the cell called the nucleus. The nucleus has a nuclear membrane which is double layered. The nuclear membrane is perforated to allow substances to enter and leave the nucleus. The nucleus contains chromosomes. Chromosomes contain hereditary information and are composed of deoxyribonucleic acid or DNA and proteins. Functional segments of DNA are called genes. The nucleus plays a major role in cell division. It also controls cell development. The fluid living content between the plasma membrane and the nucleus is called cytoplasm. The nucleus, along with the cytoplasm, makes up the living content of the cell called protoplasm. Let's take a closer look at cytoplasm. The cytoplasm is a clear, thick, jelly-like material with water, organic compounds, and organelles. The liquid part of the cytoplasm, other than the organelles, is called cytosol. Cytoplasm supports and protects the cell organelles that perform different metabolic functions. Did you know that the cell has different organelles? We will explore them now. This is the protein synthesizing site of the cell, called the endoplasmic reticulum. It is an irregular network of double tubular membrane, continuous with the nuclear membrane on the inside. There are two types of endoplasmic reticulum: rough and smooth. Rough endoplasmic reticulum has ribosomes present on it, while they are absent on smooth endoplasmic reticulum. Rough endoplasmic reticulum synthesizes proteins, while smooth endoplasmic reticulum synthesizes lipids and fats. Some of these proteins and lipids help in building the cell membrane.
Yes, Manogna, you have any doubt?
No, ma'am.
And some function as enzymes and hormones. Endoplasmic reticulum serves as channels for the transport of proteins between various regions of the cytoplasm. Do you know what these small spherical granules are? These are ribosomes found scattered in the cytoplasm or attached to the outside of the endoplasmic reticulum. These are naked and have no membrane. Ribosomes synthesize proteins. Here's another organelle. This is the site for storing and transporting proteins across the cytoplasm, called Golgi apparatus. Named after Camillo Golgi, who first described it, Golgi apparatus are stacks of flattened membrane-bound sacs or vesicles. These stacks are called cisterns. Vesicles containing soluble proteins produced by the endoplasmic reticulum fuse with dictosomes. These vesicles are transported across the cytoplasm. They fuse with the cell membrane and release the proteins outside the cell. Golgi bodies are also known for the synthesis of glycoproteins. This is the powerhouse of the